Hi guys, hope you're doing well, especially during this trying period of life. So as I said on the Facebook chat that I would make some revision videos for people just because obviously classes aren't on just now and I think that can be quite disruptive to learning, so I thought maybe this would help. So we're going to discuss pharmacology today. This was an area that I really struggled with, and hopefully by the end of this things will make a little bit more sense. So before diving into the drugs and stuff like that, I thought it would be very important to look over some of the aspects of bacteria just so you've got an understanding of what we're talking about. And I'm sure a lot of people already know this, but I just thought I'd go over it. So this is a drawing that I did of the different classifications of bacteria, and it's based upon different aspects of how they metabolize and their gram stain and particular tests that are done to classify them. And don't worry, I don't think you need to know all this. In fact, I'm pretty sure you don't. I didn't need to know this when I was doing podiatry. But yeah, hopefully you'll find this helpful. Okay, so first things first is, what do we mean when we talk about gram testing or gram positive and gram negative or gram variable? Well, this is actually a really simple test. It just is how the bacteria stain. So you add a dye to the bacteria, and then the bacteria stain different color. Gram positive tend to stain darker. Gram negative stain lighter. And the reason for that is because of their cell wall. So bacteria have a cell wall, and that's actually really important that they have a cell wall, because humans or animals or any sort of eukaryotic cell, which is basically animal cells, don't have cell walls. We just have cell membranes. But as I've just said, bacteria have both. And you have gram-positive bacteria and you have gram-negative bacteria cell walls. And as you can see, the gram-positive bacteria have a very large cell wall, and the gram-negative bacteria have a very small cell wall. In fact, this cell wall is actually sandwiched between a cell membrane and another cell membrane, whereas the gram-positive bacteria is only has one cell membrane, and that actually sits below the cell wall. So what happens is, when you gram stain, you give an injection. That looks dodgy, so we'll just wipe that away. Try and draw a syringe here. Anyway, that looks less dodgy. So you give an injection of, well, you wash them with a particular stain, and because the gram-positive cell wall is much bigger, it soaks up a lot more of this stain, and as a consequence, dyes darker, or stains darker, as I should say. Whereas a gram-negative has a smaller cell wall, and so it takes up less of the staining, and as a result, stains lighter. And that is literally the difference between gram-positive and gram-negative. I know it probably seems really simple, but I didn't know this until I started doing my other studies. And yeah, so just thought I'd summarize that there. Bacteria can then be further classified, and the next sort of main classification that you subcategorize them into is their shape, whether they're ball-like shaped or rod-like shaped, called cocci or rod, and that can be true for gram-positive and gram-negative. And then there's sort of variations in between. You have coccobacilli, which are sort of mixture of both, and then spiral. When you get past that, you start then classifying a little bit more specifically, and that can be with regards to how they cluster together or whether they chain together. You can also start looking at certain metabolic processes, so whether they're aerobic, anaerobic. And then again, with the gram-negative, they have very particular things that they seem to do, and one of the ways that you can subclassify them is whether they're lactose ferment. I almost said lactose intolerant. They're not lactose intolerant, and whether they can ferment lactose or not. But anyway, that's besides the point. Don't worry about this. What we're going to look at here is going to look at the bacteria that you've definitely heard of, called Staph aureus. Maybe you've heard of Staph epidermis. I don't know if I've got that wee arrow there drawing, but anyway. And also things like Strep pneumoniae or Strep pyogenes. These are some big bacteria that you may or may not have heard of, but you should probably have. So Staph aureus can be further divided into something called coagulase. Sorry, not Staph aureus, just the staphylococci. They can be further categorized by something called the coagulase test. And what that means is that whether they have the ability to literally promote the blood to coagulate. And the way that they do that is by activating something called fibrinogen, which is the precursor to fibrin. And as you might remember, fibrin is the thing that causes the cross-linking within the vessels and causes coagulation. And some staphylococci are able to do this and actually cause that coagulation. And Staph aureus is one of them. So Staph aureus is a Staph aureus positive, so it's coagulase positive. And you might read about Staphylococcus coagulase negative, Staphylococcus coagulase positive, and that's what they're referring to. So just remember that Staph aureus is positive. An important one, as I mentioned before, is Staph epidermis, and it's coagulase negative. And it's an important bacteria. It's one that often affects the skin and prosthetic joints. In fact, so much so with prosthetic joints that if you have someone who has one and gets an infection, you actually treat them as if they have Staph epidermis because it's so likely to be that. When you look at the streptococci, so remember the streptococci are also cocci shaped, that they are a ball, but they don't cluster. What they do is they make chains, and so they get called streptococci. Strept, I guess, meaning some sort of chain-like. These can be classified by two different classification systems, one called the hemolysis test and one the Lancefield test. I wouldn't get hung up on them, but it's just worth maybe knowing that they exist because you may hear about things such as group B strep or group A strep or hemolytic alpha or hemolytic beta. And what they refer to, well, the Lancefield, I don't really know what that refers to. It's just a classification system of something that they've put on them and has been able to subdivide them. But what the hemolysis test refers to is how it interacts with blood. And so the alpha lot, so strep pneumonia, the thing that causes your typical pneumonia, will interact with blood differently to how, for example, beta would. And I just know that off the back, beta actually completely hemolyzes your blood, and it's very dangerous. This bacteria here, strep pyogenes, is a really nasty bacteria, and it's the main one that causes strep throat, and also can cause things like necrotizing fasciitis, so it's probably worth just being familiar with it. But what you'll see here is that it is, taking strep pyogenes as an example, if you hear about things called streptococci beta hemolytic, that could mean strep pyogenes, or it could also be called group A strep, and at the same time that would mean strep pyogenes. Another famous one people have heard of is group B strep. It's important for pregnant women, and what that is really referring to is one called Streptococcus algagalactic bacteria. So just making you aware, I think it's important to be familiar with these different classification systems. It will certainly help you when you're reading around these things. Okay, so onto the microbials or the antibiotics. What I've done is I've split them down into their main constituents and how they work and how they kill the bacteria. Now there's a lot of antibiotic names here, so what I've done is in the red this is the actual antibiotic names, in green this is their like sort of subgroup, and then in blue is their main cause. I just realized as I said that I've done the complete opposite for these two down here, so probably just ignore everything I said there. But anyway, so bacteria can be killed or inhibited, i.e. you can have bacteriostatic activity by interrupting several different mechanisms on them. One of the big ones is by destroying their cell wall. Now as I mentioned, bacteria have a cell wall and humans do not. So this is a really good way to harm bacteria without particularly harming us because we don't have a cell wall. We can also interrupt their protein synthesis, and this may include their protein translation, so carrying out particular activities that make them dangerous or even interrupting their metabolic pathways. And we'll talk about that. And then finally, you can also interrupt their ability to create nucleic acid synthesis. And again, this can fall under their metabolic pathways or even just affecting the way that they transcribe or the way that they replicate. And what I've done here is I've broken down the in particular bits, which we'll talk about. I know it looks scary just now, but it's really not when you go into it all. Okay, so the big ones to talk about are a group of bacteria called a group of antibiotics called beta-lactams. Okay, and you may have heard of beta-lactams, and if you were anything like me, a bit of an idiot, I had no idea what that meant. I just knew that it was a thing. What it is it's a massive umbrella term for a whole set of different types of antibiotics. So beta-lactams, one of the things they all share in common is a particular structure within them. This isn't accurate, it might not even have a hydroxyl group, I don't know. But what they do have is they have something called a beta-lactam ring. They all share that in common, all of them have this. What that means is that they work in the same way, because this is the active portion of the beta-lactam, and I'll come on to describe how this works. So within the beta-lactam family you have penicillins, which you would have heard of, because within the penicillin family you have amoxicillin, flucloxacillin, benzopenicillin. But you also within the beta-lactam family have ones called cephalosporins, which are a very powerful type of drug, especially one called ceftriaxone, which is used in meningitis. You have ones called carbapenems. These are very important antibiotics because these are one of the few last remaining antibiotics that has very little resistance and is really used as a last line medication. So if you see a patient on meropenem, which you wouldn't really be seeing out with hospital, the person has a very horrible strain of bacteria that is resistant to pretty much everything else, and pretty much all these is resistant to, and this is the last thing. Then you have monobactams. They're not particularly used that much, and then you have a mixture, and we'll talk about them. These are quite cool, so I'll mention these. So what I'll do is in this video I'll talk specifically about beta-lactams, maybe touch on these glycopeptides. Actually, I should probably mention that here is that there's two major categories that inhibit cell wall synthesis: is the beta-lactams, which we just discussed, and glycopeptides. And within that, you have a particular drug called vancomycin, which I'm sure you've heard of, which is used for MRSA. And I also mentioned this staph epidermis here. This is a drug, vancomycin, is very good against staph epidermis, which is also coagulase negative. Okay, so we'll touch on the beta-lactams here. So how do they work? Well, beta-lactams work by having that beta-lactam ring, as I talked about. Now, they all share that in common. They will bind to the same area. On the bacterial cell wall, there is sugar molecules and protein molecules, or glycopeptides as they're called. And the sugar molecules are these things here, okay? And they are called N-acetylglucosamine and N-acetylmuronic acid, or NAG and NAM for short. And then cross-linking them, you have these proteins called oligopeptides. So the wall is literally made up like a wall. So you have your sugar molecules, and then your proteins, and then your sugar molecules, then your proteins, then your sugar molecules, then your proteins, and you get the gist, and you literally create a cell wall. Now obviously these need to be cross-linked because you have this area that's cross-linking here. There's an enzyme that does that called transpeptidase, okay? And what it does is it comes along and it joins a nice secure attachment between the oligopeptides and secures the wall. The way that beta-lactams work is that they inhibit this enzyme. And the way that they inhibit this enzyme is they bind to a particular protein, which is here, which I've shaded in, called the penicillin binding protein, which is nice and helpfully named. So that's the penicillin binding protein. And what happens is the beta-lactam ring snugly fits into that penicillin binding protein area and inhibits the transpeptidase. As a result, the cell wall cannot cross-link and it's weak. There's no cross-link in here. Okay, so I'll rub that out. What that means is that when the bacteria doesn't have a secure cell wall, lysis will occur, which I've drawn here. And what that means is the bacteria has a whole lot of proteins inside it, so it exerts an osmotic pressure. In other words, there's more solutes in here than there is around it. And as a result, water gets pulled in to it. And normally this isn't a problem because the bacteria's cell wall can hold it. It's strong enough to withstand that pressure of the water bulging within it. But when the cell wall is not working, it literally explodes. It literally pops. And just because all the water comes in and it explodes. And it releases all its content into the blood. And so one of the things that's quite interesting actually is that when you give someone antibiotics, they may actually feel a little bit worse. They may feel slightly more ill straight afterwards or shortly afterwards. Their temperature might spike a little bit. And the reason for this is because you're literally releasing all this rubbish into the bloodstream. And the immune system's coming over and taking it all up and then upregulating an immune response. But very quickly this will settle down as the bacteria is further eliminated and your immune system takes over. So one of the other things that I mentioned about the beta-lactams is you get combination drugs. And the reason you get combination drugs is because some bacteria have become quite clever and they produce stuff called beta-lactamases. And what these are is these are other enzymes which can bind to the beta-lactam ring. So you've got your beta-lactam here, and it's got its beta-lactam ring, and the bacteria produce a substance which inhibits it, and it prevents it from binding in here, and so it makes it not work. And these are little molecules called beta-lactamases. Okay, beta-lactamases. So what we can do is we can give combination drugs such as amoxicillin and clavonic acid. I don't know how you pronounce it, which is augmentin, which you may have heard of. Okay, and what the clavonic acid does is it binds to these beta-lactamases. So just redraw this. This is a beta-lactamase. It will bind to these beta-lactamases and inhibit it from binding to the penicillin or the beta-lactam ring, which will then allow the drug to carry out. So the combinations are two drugs. They are one of the beta-lactams, one of the penicillins or cephalosporins or carbamazepines, and then another drug which inhibits the carbamazepines. Sorry, getting all confused. Which inhibits the beta-lactamases. So I don't feel I've explained that too well, so I'm just going to reiterate. Bacteria can produce something which inhibits this binding to the transpeptidase enzyme. So it actually inhibits the function of the antibiotic. We can give a substance which will prevent that from happening and will allow the drug to continue to work. If you have any questions about that, I will happily answer it because I don't think I explained that terribly well. Okay, so that's beta-lactams. That's how they work. Okay, all these work exactly the same way, but they have slightly different effects on different bacteria and so are used differently. So what drugs do you use when? Well, these ones I think are the ones that you guys are most familiar with and certainly are some of the most important ones. These drugs here are used for much more severe disease. As I mentioned, ceftriaxone is used for things such as meningitis. Meropenem is more used for severe resistance. So one of the things I thought I'd talk about very quickly would be cellulitis and how you'd use it. So I'm just going to go to my notes. I think it's here. I loaded it earlier. Yep. Okay, so the cellulitis. So as you know, cellulitis is an infection that infects the subcutaneous tissue, okay, and the subcutaneous tissue only. Sorry, please ignore the rest of my notes. I'm linking it with all sorts of other things. Now, which drug do you give? Well, you give flucloxacillin, and the reason you give this is because this drug is most useful against the most likely pathogens to be causing the cellulitis, which are strep and Staph aureus, and specifically strep pyogenes, that nasty bacteria which can go on to cause all sorts of horrible things such as necrotitis and fasciitis. Now, when you give antibiotics, you're giving the antibiotics towards the most likely pathogen to be causing the issue. So as I've wrote up here, and I've just wrote down there, is that the most likely pathogens to cause cellulitis are Staph aureus or beta hemolytic strep or group A. So here, remember I was talking about that, those groups, here it is right here. So as I mentioned before, that's pyogenes. And so we need to give a drug that's going to take out both of them. And the drug that's most likely to take out both of them is flucloxacillin. So another important thing to think about is, should you give it IV or should you give it per oral or mouth? And really this is based upon how unwell the person is. Okay, so if the person isn't particularly unwell, they are just, moderately unwell, then they can probably tolerate it just being given orally. But if the person has sepsis, then you want to be given it IV. And just in case any of your exams ask you, like, why are you giving it IV, and you just say sepsis, and then it might go on to say, well, why does sepsis? Well, there's two main reasons. Number one, speed. Okay, you're giving it straight into the veins. There's no mucking around, straight in. And also you give 100% as well. So unlike when you give a drug orally, which has to then be processed, and only a certain amount of it will actually become available, i.e. the bioavailability might not be that high. Whereas when you give the drug IV, 100% goes into circulation. Number two, though, is absorption. I can never remember if it's a P or a B there, but anyway, absorption. And the reason for that is because when someone has sepsis, they have circulatory shutdown. Now it gets a little bit more complicated than just saying that they shut down. Unlike normal shock, because if they go into septic shock, unlike normal shock where you get, or hypovolemic shock where you get peripheral shutdown, in sepsis because of the release of the toxins from the bacteria, you actually get dilation, you actually get more blood going to your skin. But the point is that your circulatory shutdown will occur in the gut. So the gut blood flow will be poorer, and therefore you will not get as much, you will not absorb as much, get less into circulation, because you won't absorb as much. Sorry, I'm trying to write and speak at the same time, it's a bit of a nightmare. So when would you give IV? When the person is really unwell, when they have sepsis. And the reason you do that is because of the speed and the absorption of the drug. Now, you may have read here, I've wrote stuff like second line, Enron classification, etc. So just so you know for your exams as well, cellulitis is based upon the classification of Enron. And this is really to do with how unwell someone is. And there's 4 classifications to a person. And this is when the person's pretty well, they just got an infection on their leg. This is when they're a bit more unwell and they have a bit of systemic illness. This is when they're septic, and this is when they have neck fash. And if anybody wants to know about neck fash, I'll happily do a wee video on it. It's a really devastating infection which has a very high mortality, and I've seen it a few times, and it's unbelievably fast, and people do lose limbs. They have to get surgical resection. So anything above here, you're given IV for, absolutely. Here, you can give per oral. At number 2, Enron's classification 2 is a bit of a mixed bag. It'd be your clinical judgment if you would give that orally or not. But I just thought I'd sum that up very quickly, because obviously cellulitis is something you'll come across. Oh look, I've been spelling Enron wrong. Never mind. It's something that's very important to know about your clinical application of antibiotics. But the main thing to take away from this is that for cellulitis, you want to give flucloxacillin. That's your number 1 drug. And as I mentioned here, your second line would be doxycycline, and that's really for people who have allergies to penicillin. And then if the person becomes very unwell, then you can move to IV medication. So that's the beginning of the pharmacology, and I've covered the beta-lactams, which is probably one of the bigger subjects and is really important. And after this, if you want, we can go through things like erythromycin and their classifications and particular things that you need to watch out for in them, and go through to folate inhibitors. I know all this sort of stuff looks really daunting, but you don't need to know it. It's just to help, with your studies. Okay, thanks.